Today, we are learning oxidative phosphorylation in IB Biology HL, how electron flow builds a proton gradient, and how that gradient drives ATP production. First, do not think of oxygen as making ATP directly. In oxidative phosphorylation, electron flow is what builds the proton gradient. Electrons move through the electron transport chain. Their energy pumps protons across the inner mitochondrial membrane. At night, and fed to donate high-energy electrons. As those electrons pass along the chain, energy is released and used to pump H+ across the membrane. Oxygen acts as the final electron acceptor. That keeps electron flow going, so the proton gradient can be built. So in an exam answer, link oxygen to continued electron flow and proton pumping, not to ATP production directly. Now the gradient has to be converted into ATP. That process is called chemiosmosis. The proton gradient stores potential energy. H+ builds up outside the matrix in the intermembrane space. That difference in proton concentration stores energy. The protons then flow back through ATP synthase down their concentration gradient. APP synthase uses that flow to phosphorylate ADP into ATP. So the key wording is ATP synthase uses proton flow, not electron flow, to make ATP. Here is the clean version to remember. The electrons enter the chain from NADH and FADH2. Their energy pumps protons. That builds a proton gradient. The gradient drives ATP synthase, and ATP is made from ADP and inorganic phosphate. The memory line is electrons build the gradient drives ATP. At exam answers, use final electron acceptor, mention the proton gradient, and link the answer to ATP synthase. Question A is a real back-end preview question about glycolysis. Glucose is converted to pyruvate during glycolysis. First, label P where phosphorylation occurs. Then label O where oxidation occurs. Finally, state where glycolysis takes place in the cell. For the answer, P is the phosphorylation step near the start of glycolysis. O is where triose phosphate is oxidized before pyruvate forms. And the location is the cytoplasm or cytosol. Question B shows the last step of anaerobic respiration in human skeletal muscle cells. The question asks for the purpose of this reaction. Read the options carefully. The key is what the lactate reaction restores. Option A says it allows energy release from glucose breakdown to continue. Option B is about a hydrogen ion gradient for ATP synthesis. Option C gives electrons to the electron transport chain. Option D says it reduces carbon compounds in the absence of oxygen. The correct answer is A. This reaction regenerates NAD, so glycolysis can continue releasing energy when oxygen is not available. The hydrogen ion gradient option belongs to oxidative phosphorylation, not lactate formation. Question C is a 3-mark explain question. All cells require a supply of energy. Explain the properties of ATP that make it useful for distributing energy within cells. For this kind of answer, build 3 mark points. First, name a property of ATP. Second, explain how energy is released. Third, link ATP to a clear cell process. A strong answer is ATP is a small soluble energy carrier, so it can move around the cell. ATP hydrolysis releases usable energy. ATP can also phosphorylate molecules, helping drive processes such as active transport, movement, or biosynthesis. So the exam habit is simple. Use the question diagram, avoid the distractor, and turn each biology phrase into a clear mark point. For scoring tips, keep the chain clear. First, oxygen is the final electron acceptor. Second, electron flow powers proton pumping across the inner mitochondrial membrane. Third, the proton gradient drives ATP synthase. Fourth, link ADP and inorganic phosphate to ATP production. Remember, oxygen does not make ATP directly. It keeps the electron transport chain moving. Keep practicing with real exam questions on EduNinja. You can work topic by topic, check step-by-step solutions, build smart papers, and review notes when you need them.